All right, here we are to talk about enzymes, and hopefully you remember that an enzyme is a protein. Anytime you see an enzyme or are dealing with an enzyme, you always have to start thinking of the protein properties. So, one thing that is sort of a clue for you to look for is this ending in ASE, that suffix. There are a couple exceptions, but every word in bio that ends in ASE is going to be considered an enzyme. Big thing to understand is that while enzymes take part in a reaction, they themselves always stay the same. So they're going to change something called the substrate into a product, but the enzyme is always going to remain the same, and we'll see that in a couple pictures as we move forward. Enzymes are going to act as catalysts. Catalysts are items that are going to make a reaction happen faster. And the way they do that is they work on something called activation energy. Activation energy is the amount of energy it takes to get a reaction going. Now, a reaction might take place without an enzyme, but if you add an enzyme, that reaction is going to take place a lot quicker and you're going to get to your product faster, which inside a cell, efficiency is key. So enzymes are really important in maintaining efficiency. There's a chance without an enzyme the reaction doesn't happen at all. So, I mean, these things are really important, especially if it's a reaction that needs to happen for the cell to maintain function. Here's a graph of what happens with all reactions. We have sort of our stable reactant. Reactant's the thing that's going to be going into the chemical reaction. Excuse me. And then in order for the reaction to happen, you have to have an increase in energy before you have this decrease in energy, which is the release that results in the products. So what an enzyme is going to do is going to take this part and lower it so that this reaction can just go kaboom and go a little bit quicker to get to your products. So if we compare these two graphs, we see an uncatalyzed reaction, which is a reaction without an enzyme, and a catalyzed reaction, which is one happening with an enzyme. And you'll notice the activation energy, which is right here, is a lot lower than the one with the enzyme. But look at the bottom, the products. Is there any change in the amount of products? No, that all stays the same. So all these do is help you get from point A to point B faster. And that's a big thing with enzymes, is that they speed things up so that you can get moving a lot quicker. So this is just sort of a summary of what I was just saying, that the enzymes work to get you to the products a lot quicker. Now let's see how this goes on. And if you want an idea of how quick it can go, we're talking about 100 to 1,000 substrates per second. So I mean, we're talking really quick stuff going on here, which inside a cell, efficiency is king. So when we talk about enzyme reactions, we always start with two things. We start with an enzyme, which is the protein that's going to make the reaction happen, and a substrate, which is the thing that's going to need to change. When those two things join together, they form what's called an enzyme substrate complex. When the enzyme substrate complex attaches, the enzyme puts something called a strain onto the substrate, and then after the strain, one of two things happens to the substrate. It either breaks down or it gets put together to make something a little bit bigger. And then that's going to turn into a product which is different than the substrate, and then the enzyme remains unchanged. It's the same as it was before the reaction. So the enzyme is going to stay the same throughout this entire process. The substrate is the only thing that's going to get changed, and it gets turned into the product. Now, when we look at enzymes, there's a couple parts. We have enzyme here with these spaces. Remember, proteins fold all over themselves, and those folds cause a three-dimensional structure. And in that three-dimensional structure, there's spaces that can be considered what we call active sites. Active sites are places where the substrate can hook into. When the substrate and the enzyme hook in, now we have our enzyme substrate complex. And if we have water going into this reaction and we see things breaking down, hopefully you recognize that this is a hydrolysis reaction specifically. We could also have a condensation reaction where water would come out and two separate things would join together. But we have the enzyme substrate complex, it puts some strain on it, substrate gets converted, substrate gets released, and then the enzyme's open to do more work. And it's just a big cycle because remember that enzyme never gets changed. So what I was talking about where the substrate and the product or the substrate hooks in with the enzyme puts on something called inducing strain. Inducing strain is this idea that the different three-dimensional sort of holes in the enzyme hook onto the substrate, and as they do that, the enzyme sort of changes shape a little bit and sort of bends the bonds. If you remember from class, we said there's a lot of energy held in bonds, and bonds are hard to make and hard to break. So the enzyme sort of works on those bonds and gets things ready for the conversion from substrate to product. This idea that the enzyme sort of wraps around the substrate to get onto it is induced fit. And induced fit is kind of like a kid sitting into a beanbag. The beanbag is sort of fluffy, and then as soon as you sit in, it sort of wraps around you. So that's sort of what we're talking about with induced fit, where the beanbag would be acting as the enzyme in this picture. Now, when we look at this graph, we are seeing a reaction with an enzyme, and we're seeing a reaction without an enzyme. Keep in mind, without an enzyme, there's a chance this reaction never happens, but look at the speed of these. This one shoots up really quick, and then it sort of plateaus off. It plateaus off at that maximum rate. The maximum rate occurs when there's enough substrate concentration and enough enzyme molecules to be occupied. So it's going to top out eventually, but look at it without the enzyme. It never even gets close to that maximum rate. You have to have that enzyme to make the reactions go really quickly. So that's where these things are really important in biological processes. Sometimes enzymes need a little bit of help, and that help is something called a coenzyme, which co means with, and it just goes with the enzyme. It's just a small molecule that's going to bind to the active site and just sort of help things along. So every so often you'll have a coenzyme. We'll talk about that stuff later in the year because there are some things. Acetyl-CoA is an enzyme that we'll talk about in the CM pathway and other coenzymes that we'll get to. When a reaction happens, one thing you do need to know, every substrate is specific to an enzyme. So there's thousands of different enzymes within our cells, and the enzymes are proteins, which are made from the code in the DNA. So the DNA inside each cell sort of determines which enzymes are made at which time, and a whole bunch of cellular processes that we'll start getting to as we move forward in the year. We're going to hear a lot about these enzymes. So this is something that is pretty important for what we're doing in this class throughout the course of the year. I went too far. Now, enzymes need regulation. We can't just have enzymes going all crazy and just going back and forth, back and forth, back and forth, and just making things whenever we need it. So we need a way to turn them both on and turn them off. Inhibitors are going to be something that stop the enzyme from working. They inhibit it. So there's a couple different kinds of inhibitors that we can have. We can have an irreversible inhibitor, where it's something that's going to bind to the enzyme to basically turn it off forever. DIPF is an example your book gives, and it inactivates acetylcholinesterase. Acetylcholinesterase is something that breaks down a product called acetylcholine inside your nerve cells, which acetylcholine is a neurotransmitter that makes nerve cells transmit information. Well, if you stop acetylcholinesterase, that's going to make your acetylcholine stimulate the nerves constantly, which can lead to paralysis, which is why it's a really good insecticide, something that can kill bugs really quick. Also, we have reversible inhibitors, and this is more what we deal with in our class. Reversible inhibitors come in two forms. A competitive inhibitor is going to sit inside the active site of the enzyme. If you remember, the enzyme had those sort of cutouts in it. Well, a competitive inhibitor is going to compete with the substrate for a spot in that active site, and it's going to block it. Kind of like something if you put a cover on top of a basketball hoop and people were trying to shoot at the basket, they wouldn't be able to get the ball in the hoop. That would sort of be how a competitive inhibitor would work. A non-competitive inhibitor is going to attach somewhere else besides the active site. But when it attaches on the enzyme elsewhere, it's going to cause a change in the shape of the protein, which is going to change the shape of the active sites and not allow things to get in. Non-competitive inhibitors are also called allosteric inhibitors. That's an important word that you're going to be hearing a lot of. Allosteric inhibitors are always going to be non-competitive. So we'll sort of see examples of that. Here's a picture of a competitive inhibitor. I have my enzyme right here, this brown thing with kind of these three little fingers. The competitive inhibitor is going to fit inside the active site. Here's a substrate. Can't get in there. So you can't interact with the enzyme. If the substrate doesn't interact with the enzyme, the reaction never takes place. With a non-competitive inhibitor, I still have my enzyme. I got my three little fingers here. This non-competitive inhibitor or allosteric inhibitor is going to hook in away from the active sites. And look what happens to the active site when it hooks in. It closes up. Can't get in again. So either way, the result's the same. It's just a matter of how you get from point A to point B, from open active sites to closed active sites. When we talk about all these enzyme catalyzed reactions, these things are what we consider metabolic pathways. Metabolic pathways are a series of things that we're going to talk about that are catalyzed by enzymes. And there are these reactions that take place within the cell, and it's sort of like pulling a trigger on a gun. Once you pull a trigger on a gun, then the hammer is going to hit the primer. The primer is going to ignite, that's going to hit the gunpowder. The gunpowder is going to ignite, and that's going to propel the bullet out of the gun. Well, that's sort of how a metabolic pathway starts. Once one thing happens, that leads to the next thing, the next thing, the next thing. It's kind of like a chain reaction. And that initial reaction is called our commitment step because we have to get everything going. That's like you pulling the trigger on the actual gun. When the reactions are happening, we have to get that ball rolling, and sometimes we want it to stop. Sometimes we'll hit a point where we want the reaction to stop, but we want it to stop only at a specific time when enough products have been made. When the number of products determines how the reaction is regulated, we're going to call that feedback inhibition, because feedback from the products is going to inhibit the enzyme. And we'll get to that stuff a lot later in the year. I just sort of want you to get you familiar with these terms right now. The last thing we'll talk about are things that can change a way an enzyme works. And if you remember from proteins, there's something called denaturing of proteins. The denaturing of proteins happens when the secondary, tertiary structure of the protein degrade due to changes in a couple things. And two things that we said change it really well are temperature and pH. And enzymes can absolutely get denatured because they are proteins. So there's specific temperatures and pHs that we call our optimal temperature in this graph and our optimal pH. And if you look at the pH one, each enzyme has a different optimal pH. Well, inside your body, there's different pHs throughout all the different places in your body. This one, pepsin's inside your stomach. Your stomach is a very acidic area, so its optimal pH is around a 2. Well, other places in your body aren't that acidic. Nothing else would survive there. So chymotrypsin, its optimal pH is around a 7.5. Arginase, the optimal temperatures or optimal pH, is around a 9.8. When we look at temperature, notice this graph. It goes up slowly, goes up slowly, goes up slowly. We hit our optimal temperature. And then look what happens after you get past that optimal temperature. Boom, crashes really big. That optimal temperature, what do you think that is for humans, for most enzymes? I'm probably guessing it's going to be around 98.6 degrees or 37 degrees Celsius. That's your normal body temperature. Well, when you get a fever, if you get too hot, and you guys read the article about hyperthermia and exertional heat stroke, well, when that happens, your body temperature gets up into this range. Look what's happening to enzymes over here. Their function is crashing. And if that's the case, well, your enzymes don't work, then those pathways don't work. If the pathways don't work, your cells don't make the products they need to maintain function. And if that doesn't happen, then you can die, which is why heat stroke is a really big issue. So we got optimal temperatures graph is slightly different than optimal pH, but it's something that can change it because denatured enzymes do not work. They don't make the products. That's where we're going to stop with enzymes. That stuff's all from 3.3. And go there and check out anything else you need. Feel free to email me or talk to me in class about any questions. Have a good day.